All right, let's just jump right into this explainer. Today, we're acting as computational detectives, and we're trying to crack a massive biological mystery. We're taking on one of the absolute most ambitious goals in structural bioinformatics, predicting a complex, three-dimensional protein structure from nothing but a simple, one-dimensional sequence of amino acids.
So here's the crucial question. Why is simulating biology inside a computer so incredibly difficult compared to just running an experiment in a physical lab? I mean, today we can easily generate computational protein models. That's not the hard part. The surprisingly tough part is figuring out which of those models is actually closest to reality. We calculate the model energies, and we basically gamble that the lowest energy is the most realistic. But the limited accuracy of today's energy functions, that's exactly what's holding us back.
And that struggle is perfectly captured by this idea of the last mile of the protein folding problem. It hits the nail on the head regarding our central conflict. See, when our predicted model gets really close to the real structure, the demands on our computational energy functions become almost impossibly high. We literally have to fix these microscopic, tiny errors to achieve experimental accuracy. And crossing that final last mile, that is easily the hardest part of the entire journey.
Because ultimately, the goal here is to understand these actual, functioning 3D molecular machines. We want to go straight from a basic sequence blueprint to the incredibly complex folded proteins that actually do the heavy lifting inside living cells. If we can get this right computationally, we can literally skip years of grueling laboratory crystallography and NMR spectroscopy. And the stakes here? They are absolutely massive. This isn't just some academic exercise. Solving these structures is the literal first step to designing rational drugs and fixing devastating human conditions. By modeling specific mutated proteins, we can trace the structural roots of diseases right down to the atom. For instance, studying the DJ1 protein gives us critical insights into Parkinsonism, while looking at mutations in gamma-actin-1 can actually help us understand and maybe even treat hereditary hearing loss.
Now, our go-to toolkit for solving this whole puzzle is called homology modeling. It relies on this amazing evolutionary quirk. The structure of a protein actually changes way slower than its sequence. What that means is that similar amino acid sequences practically always fold into identical 3D structures. So if we know the structure of sequence A and sequence B is highly similar, well, we essentially already have the blueprint for sequence B.
So, part 1, finding the right template. The detective's blueprint.
When we're hunting for these templates, our safety actually depends on a pretty strict mathematical curve. Basically, as long as the length of our sequence and the percentage of identical residues fall in that upper safe zone, we have a highly reliable template. We're virtually guaranteed a similar structure. But the moment that percentage drops below the curve, we enter some incredibly risky territory.
It's the classic contrast between a safe, high-identity match and what structural biologists quite literally call the twilight zone. Sounds dramatic, right? But in the safe zone, our automated alignment algorithms work beautifully. You can confidently copy the backbone. But in the twilight zone, those automated matching algorithms completely fall apart. The alignment becomes prone to these massive errors, and you desperately need deep human expertise just to salvage it.
To really understand why those automated alignments fail, think about a pathological alignment problem. Imagine trying to align sequence A with sequence B. Just looking at them, they seem completely unrelated. You physically cannot align them without introducing a third sequence, sequence C, which acts as an evolutionary bridge connecting the two. This is exactly why multiple sequence alignments, where we compare whole families of related proteins, are absolutely vital to finding the right match.
Okay, so step 1, we recognize our template. Step 2, we carefully correct our alignment using the 3D context. We're basically making sure we aren't accidentally shoving a water-loving amino acid deep inside a hydrophobic core. Once that's sorted, we move to step 3, generating the backbone. And for most of the model, we can do this pretty easily, just by copying the match coordinates directly over from our template structure.
Moving right along to part 2, modeling loops and branches, reconstructing the missing pieces.
Step 4 is where things get notoriously difficult. When our target sequence has insertions or deletions compared to the template, we have to actually shift the backbone and build entirely new surface loops to bridge those gaps. Why is it so hard? Because surface loops are incredibly flexible. They get pushed around by bulky side chains underneath them, and they warp to form crystal contacts. Honestly, it remains one of the big, unsolved problems in homology modeling.
And then comes step 5, which is another massive challenge: predicting the exact 3D orientation of the side chain branches, also known as rotamers. Think of it like this. The backbone is the spine of the protein, but the side chains are the branches that actually give it its function. We have to figure out exactly how these branches twist and fold themselves into the available space.
Now, if we just tried to blindly guess every single combination of these rotamers, we'd hit a mathematical brick wall. I'm talking a full-blown combinatorial explosion. Even with just 100 residues averaging 5 rotamers each, the possible combinations are 5 to the power of 100. Let me put that in perspective. That's a 1 followed by 70 zeros. It is mathematically impossible for any computer, or even nature itself, to test every single path.
So how do our computational detectives get around this impossible math? Well, we use a secret weapon, position-specific rotamer libraries. It turns out the physical shape of the protein backbone naturally limits the search space. Certain bends in the backbone strongly favor specific side chain twists to allow for hydrogen bonds or tight packing. By using the backbone as an anchor, we can drastically shrink that 5 to the power of 100 down to a puzzle we can actually manage.
Which brings us to part 3, conquering the last mile, the final optimization battle.
This phase really exposes a brutal chicken and egg scenario. Think about it. To predict the perfect side chain placement, you really need a flawless backbone. But to get a flawless backbone, you need perfect side chain packing to shape it. So you're stuck in this continuous loop of predicting rotamers, shifting the backbone, predicting rotamers again, and just really hoping it converges on reality.
And when we try to minimize the energy of these models, the algorithms we use make all the difference. Classic force fields completely fail this last mile. Sure, they might fix a big, obvious error, like 2 atoms crashing into each other. But they accumulate all these tiny little errors, eventually guiding the model completely away from the true target. That is exactly why we desperately need self-parameterizing force fields, smart algorithms that literally learn and adjust their own rules during the minimization process, inching us constantly closer to true experimental accuracy.
But here's the reality. Even with the absolute best force fields, every single computational model has errors. There's just no escaping it. So, step 7, our final act as computational detectives, is rigorous, intense model validation. We have to hunt down where those errors are likely hiding before we can even think about using the model for drug design.
We're basically cross-examining our structural case. We check the bond lengths, the angles, we run force field energy checks. We're asking, are the water-loving polar residues actually sitting on the outside? Are the hydrophobic ones properly hidden inside? But, and this is a big but, we have to be incredibly careful here. Why? Because a perfectly minimized structure with 0 bumps and fantastic energy scores can sometimes still be a completely misfolded protein.
When it all finally comes together, though, the end result is genuinely awe-inspiring. We're talking about the sheer, wild complexity of a fully simulated protein environment, completely submerged in water molecules, with all the physical and entropic forces properly accounted for. Getting this right computationally is an absolute monument to modern bioinformatics.
I really want to leave you with this final thought. We are currently right in the thick of fighting through that last mile of protein folding. But if we can fully master this process inside a computer, if we can accurately simulate the very machinery of life from just a simple string of letters, the potential to unlock the secrets of human disease, to design revolutionary new medicines, and to understand biology at its most fundamental level, well, it's truly limitless.